Hello and welcome back to Excelsior Academy. So in the previous lecture, we talked about different stages of the mitosis, and in this lecture, we are going to talk about the meiosis. So already it is very much similar to the mitosis. The only difference is that meiosis is also known as reductional division. So here, if a organism is diploid, it gets replicated, becomes 4n, and after the reductional division, what happens is at the end you get the haploid cell. That is n you get. So this is what we're going to learn today. And this is, again, I can say that this all we have learned in our undergraduate level also, during the graduation level also. So this is just like a revision for you guys. So let's begin today's lecture. So meiosis. So meiosis is a specialized type of cell division that reduces the chromosome number by half. So it means what? In the mitosis, we talked about the 2n. That is when we say it is diploid, right? Now, in the case of meiosis, what happens? It becomes n. So these are called then haploid cells. So mainly your germline cells are the haploid cells, which are responsible for the fertilization, which takes place in the future. Okay. So what happens is in this, it reduces the chromosome number by half, that is creating 4 haploid cells. So overall, what we are going to learn in this lecture is that, and then you have got an organism diploid. So this becomes 4n after the duplication, and finally what you get is the haploid. So that's why you get 4n haploid cells. So this is what we're going to learn today in detail. So meiosis is a feature of sexually reproducing single-celled and multicellular organism. In meiosis, DNA replication is followed by 2 rounds of cell division. So this is the DNA replication followed by the 2 rounds of cell division. So that means first it becomes 2n, and then further it gives you the NN. So the cell division of meiosis is divided into 2 categories, meiosis 1 and meiosis 2. So let's study about meiosis. So of course, like how in the case of mitosis we talked about the interphase, here also the interphase is present. So nuclear material gets double in this phase. So already we know in the interphase, they are talking about the S phase. And the central replication also takes place in the interphase. Now the vesicle fuse with each other at the center of the phragmoblast and develops into a cell plate. So this is what happens. So you have got the cell, and in the center, this phragmoblast is there, which actually starts dividing, and finally what you get is 2 daughter cells. Okay. Now interphase has 3 phases. So this already we know, it is the G phases, S phase and G2 phase, G1, S and G2 phase. Now G1 phase is where the cell synthesizes all proteins which is required for the growth and DNA replication, and then only after the G1 checkpoint the cell will enter the S phase. Now in the S phase the DNA replication takes place. Now, in case of meiosis, G2 phase is absent, and the interphase is followed by meiosis 1 and meiosis 2, since we are talking over here about the reductional division, right? Because here 2 times the division is required. So you have got the cell. You can see that the nuclear envelope starts disappearing, as you know that it is happening, and then the central plates are getting doubled over here, right? Okay. So meiosis 1, so it is called the reduction division over here, right? And meiosis 1 is the segregation of homologous chromosome. Meiosis 1 produce haploid cell. That is, in chromosome, we have got 23 chromosomes in human. Now, which each contains chromatid pairs N or 2N it is actually. Thus, the ploid is reduced from diploid to haploid. Thus, meiosis 1 is called the reduction division. Nuclear membrane fragmentation also takes place in this stage. Now, we already, as we talked about that, in the prophase, the nuclear fragmentation happens. This is very similar to your mitosis. So guys, whenever you're studying about the mitosis and meiosis, make your notes and make into a tabular form in such a way that what are the similarities and what are the differences between the mitosis and meiosis. Okay, so make table and then only study. So prophase 1. So early prophase have got the 5 stages. So again, so we know that in the mitosis you had 4 stages: prophase, metaphase, anaphase, and telophase. Now here we are going to talk about the prophase 1, which has got another different 5 stages which is taking place. Okay, so it is the longest phase of meiosis. Pairing of the homologous chromosome takes place in prophase. Now what do you mean by pairing of homologous chromosome? So we know that in the reproduction or in the fertilization, you get n number of chromosomes from father and you get n number of chromosomes from the mother. Okay, now what happens is, for example, let us talk about in the case of human, you get 23 pairs from here and 23 pairs from here. Now these 23 pairs, sorry, 23 chromosomes from father, similarly 23 chromosomes from the mother. Okay, so now homologous chromosome means the identical chromosomes which are present from the father and mother are going to pair up. Okay, so if there's a small one, so there was small will be pairing up. So that is called the homologous chromosome pairing up. Now this is taking place in the prophase 1. Please remember. Okay, so homologous chromosome pairing up is taking place in the prophase 1. Now the paired and the replicated chromosome are called the bivalent or the tetrads, which has 2 chromosomes and 4 chromatids. So how does it look like? It looks like this. Okay, so it has got the 4 chromatids. Okay, so this is called the homologous pairing up. So 2 chromatids over here and 2 chromatids over here. So 2 chromosomes total, we have got 4 chromatids now. So this is called the homologous pairing up. Now, this is the first stage of prophase 1, which is called leptotene. Okay. Now, in this stage, chromatin, the fiber uncoiling takes place and condensation of these duplicated chromosomes gets started. So remember, in the mitosis also, we have talked about the chromatin condensation. So it is similar where you're going to talk about the cohescence and the condensance. Okay. So each homologous pair starts to arrange in a lengthwise manner along each other. Why? Because it has to pair based on the homologous. So the similar chromosomes which are present in the mother and father, they will come and pair up. So what is happening now? The chromosomes are getting condensed. And in the leptotene nuclear envelope, it's getting more and more fragmented. Now, second stage of prophase 1 is called zygotene. So just now what we talked about was leptotene, now zygotene. Now, in this phase, pairing of homologous chromosomes occur. This is called synapsis. And this leads to form a synaptonemal complex. And the homologous pair is called the bivalent. Now, this is also very important. Please remember. So, in prophase 1, okay, so if suppose a question comes like homologous repairing or homologous chromosome pairing happens when? So your answer should be meiosis 1, prophase 1, zygotene. Okay, now this is the perfect answer. So that is in the meiosis 1, again in the meiosis 1, prophase 1, and then the zygotene. So where the correct pairing of homologous chromosome is happening. Now what is synapsis? So what is synapsis? So if you have got one pair of chromosome from mother and you have got another pair from the father. Okay, let us say this is from father and this is from mother. Now, what is synapsis? Synapsis means that this is getting crossing over. So, what will happen during this? Because of this crossover, it will look like something like this. Sorry. So, something like this it will look. So here, this portion of blue will go here and this much portion of green will go over here, right? Now, this particular thing is called, also called homologous recombination. Okay, now this is important. Now, why? Because this recombination brings about the uniqueness in the offspring. If suppose you get as it is, so you can say that 50% you're like mother, 50% you're like father, right? When this recombination happens, that is the time or that is the chance where the offspring is going to have something unique, a difference is there, right? So this is also very important. And that's how the child brings up, you know, its own, qualities. Okay. The third stage The third stage of the meiosis one, prophase one, is packeting. So here crossing over takes place. So this is what over there it was the repairing happening, and in the packeting the crossing over is taking place. So the non-sister chromatids of homologous chromosome exchange their parts. In this phase, synapsis gets complete. So in the zygotine, in the previous one, you can see that the pairing is happening. You can see over here the pairing is happening and the synapsis begins. Now, whereas in the case of packetine, that synapsis is getting over. Okay. Now chromosome gets more condensed and now they are called tetrad. Okay. In this phase, crossing over occur, that is the physical exchange of genetic material. Like I have shown you, like how that small piece of one chromosome is getting shifted to the another. Now this is happening between non-sister chromosome. Now what does it mean? Again, so these two are called sister chromosomes. Okay. Now if you have got the another, so it is happening between this and this. So that is called your non-sister chromosomes. They are called sister, and pink and blue will be called as non-sister chromosomes. So now where this crossing over is taking place, it is called chiasmata. So synapsis is complete, crossing over occurs in the packeting stage of the prophase 1 of meiosis 1. Okay, now the fourth stage is called the diplotin. So what all we have studied? Leptotin, zygotine, packetin, and diplotin now. So this is the fourth stage of the 5 stages of the prophase 1 of meiosis 1. So chiasmata formation takes place in this stage, which represents the site of crossing over. So chiasmata provides evidence of crossing over. Okay. So now what do you see over here? That synaptonemal complex starts disappearing and chiasma will be starting visible. Okay. Now, the last stage is the dikinesis. Now, it is the final stage of the prophase 1 of meiosis 1. In this phase, the shortening and thickening of the paired chromosome takes place. So, what does it mean? See, if the pairing is happening like this, okay. So, let us imagine that these two are the homologous chromosome. Now, what is happening is that they start getting more and more condensed. Okay, so this is happening in the dikinesis. So that is what the meaning is, that shortening and thickening of the paired chromosome. So this is also again similar to mitosis. Nuclear membrane disappears, chromosome condense further. So you know what is that protein called? It is called condensins. Okay. Now this condensation makes the 4 chromatids of synaptonemal complex disappear in this phase, and now homologous chromosome movement occurs. Now why this movement is required? Because it has to enter the next phase of the cells, phase of the meiosis. Okay. Now in the late prophase, microtubule formation restores in the prophase. So you know microtubule formation means they are talking about the spindle fibers, which will go and bind to the kinetochores of the chromosomes. The centrosome gets split to form 2 centriole, which moves towards the opposite pole, and asteroids formation from the central to get started. And thus, in the late prophase, spindle apparatus formation gets started. Now, why spindle apparatus? Because you know that it is going to bind to the kinetochores of the chromosome so that they can easily push the chromosomes into the opposite direction. Okay. Now the nucleus gets disappeared and chiasmata are visible. So this is the last stage of the prophase 1 called dikinesis. Now, so now the prophase 1 is completed. Okay. Please remember we are talking about the meiosis 1 now. Now, second stage, what is it's called metaphase 1. So again, it is similar to the metaphase of the mitosis, what we have learned already. So in the metaphase, what happens is your centriole reach at the opposite pole and the spindle apparatus gets established. So spindle apparatus is your microtubule proteins. Okay. Now at the pole, centriole give rise to the astromicrotubules which remains in the cortex. So astromicrotubules are the ones to which none of the chromosomes will be attached. Okay. Remember that. So they are just, these are your astral chromosomes. Astral spindle fibers. At the pole, central give rise to 3 types of microtubule. Astral, to which nothing is attached. It remains in the cell cortex, these ones. Okay. And then central also give rise to kinetochore microtubules, which gets attached to the homologous chromosome pair at the kinetochore. Again, very much similar. This is something which we have already learned in the metaphase of the mitosis. And then the third one is called the polar microtubule. And these microtubules are helping in the movement of the chromosome with the help of the microtubule motor proteins. Now, what are those microtubule motor proteins? Remember, we talked about the kinesin, which moves in the plus direction, and we have got the dynein, which moves in the negative direction. Now, with the help of these motor proteins, the chromosomes are pushed away from the center towards the opposite poles. And now these motor proteins work on the polar microtubules. So astral microtubules are present on the cell cortex. These polar microtubules are working like in roadways onto which these microtubules are working. One second. Okay, so sorry for the disturbance. So back to the class. Okay, so what I was saying was, so with the help of these polar microtubules, these motor proteins walk on these and they push the chromosomes towards the opposite direction. Okay, so this already I have mentioned in the mitosis stages, the previous lecture, that it is based on the frictional force mechanism. Okay, so if you understand this, you can also understand how these motor proteins are actually carrying the chromosomes. Okay, so one more microtubule arises by the centriole called the polar, and it is a third type of microtubule. And these polar microtubules from both sides overlap each other and help in chromosomal movement. So I hope this is clear. This is actually very interesting and also important to understand. Now, next is the anaphase. Again, so from now onwards, whatever we are going to talk about is very much similar to the mitosis. Okay. So in this phase, homologous pair gets split up and they move into the opposite direction. Okay. And thus it is called reductional. So here what is happening? It is getting divided into half now. Okay. Now in late anaphase, homologous pair that is having the sister chromatids will reach to the opposite pole now. Now again, similar to the mitosis, telophase 1 is there. So now each centriole represents a centrosome and it will be moved towards the opposite direction. You can see two are there, okay? Now the chromosome gets converted into chromatin, fiber as well as nuclear membrane gets assembled around the chromatin, and finally what will happen? The entire chromosomes will be forming into the chromatin now, okay? Nucleolus reappears in this stage. Cleavage furrow formation gets started in the phase which leads to cytokinesis. See, this is one you can see. Okay, so cleavage furrow is made and finally the cell is divided into two. And the cytoplasm is also gets divided among the two daughter cells. So now cleavage furrow formed by the actin myosin II, which is a motor protein, and derive the cytokinesis process. So this was all totally similar to the mitosis, what we have learned earlier. Okay, now interkinesis. This is something which haven't discussed before. So interface between the meiosis 1 and 2 is called the interkinesis. Okay, now what happens here is that none of the nuclear material is getting synthesized again. So whatever is present, that itself is getting divided. So you can see now two daughter cells are there and both are going to be similar. So you can see plasma membrane is there. Now the centrosome was there, which gets divided into two now, two central pairs. The chromatin is there, nucleus is there, nuclear envelope is there. So these two you will see that those are same. Now again I repeat, interkinesis is the phase between the meiosis 1 and 2. Now let's talk about the meiosis 2. So it is a separation division and it is similar, very much similar to the mitosis. So here what is happening is in meiosis 2, the segregation of sister chromatids is happening. Now the meiosis 2 is nothing but actually your mitosis now. Okay. So in this stage, chromatin, again, the fiber condensation starts again. Nuclear membrane will again disappear. The centrosome will split into two and they will reach into the opposite pole. So now if you see that both the figures are actually going to be same, since here now we have got the two daughter cells, right? So your centrals moves towards the opposite pole and again the nucleus will get disappear now. So metaphase 2, here the centrioles you have reached the opposite sides and again the three types of microtubule will be produced, that is your astropolar and the kinetochore ones. Kinetochore will go and bind to these centromere and they will pull the sister chromatids into the opposite direction. So finally what will happen? So these sister chromatids now, these sister chromatids will be pushed into the different direction. Okay. So again same. Now, so what happens in the anaphase? The sister chromatids are getting separated, reaching the opposite pole. Now in the telophase 2, the chromosomes start getting into the chromatin fibers, and you can see that the cleavage furrow is again started formation with the help of the actin and myosin 2. And finally, at the end of this, what you're going to see is that from each cell, you're going to get the another two, right? Another two daughter cells. So total, you're going to have the 4 haploid daughter nuclei through the completion of the second cytokinesis. Okay. So that's all about the meiosis 2. So from here you can see that just try to make a table so that you don't remember, so that you don't forget. Okay, that see, make a table like this where you have got, first you talk about the similarities between the mitosis and the meiosis. So you can talk about the mitosis and meiosis, what are the similarities, and in the second table you can talk about the differences. So you can talk about the differences, for example, as mitosis is having only one, whereas meiosis two stages are there, meiosis 1 and 2. And if you talk about the similarities, meiosis 2 and mitosis are completely similar. So at the end of mitosis you get two diploid cells, whereas in the case of meiosis you get 4 haploid cells. So this is how, if you make the table, it will be very easy for you to remember the differences and the different stages of the mitosis and meiosis. Okay, so I hope this lecture was helpful to you guys, and with this I would like to say thank you. If any doubts are there, please message me, or you can comment me in the YouTube channel, or you can also ask me in the Telegram channel. Thank you.